Thank you so much for all the interesting talks and posters so far, and thanks for your attention in these last few minutes. Um, I'm going to be talking about, yes, this method to use multimodal data to infer cell-cell communication and signaling pathways. So I like to think of the problem of multimodal data as sort of analogous to the parable of the blind man and the elephant. So if you look at any one aspect of what's going on in the cell, the proteins, the transcripts, the epigenetics, everything like that, you're going to see some true information, but you're going to miss like the whole picture unless you bring these different modalities together. So it's no surprise that this is something that's a really interesting problem that a lot of people have been working on. Some of these methods have been mentioned today already, especially I think I heard some talk about cell phone DV or multi-VI. So there's methods out there which can integrate multi-omic data, and then there's also some methods which are doing which are addressing another interesting problem that I find really useful, which is inferring cell-cell communication. But none of these methods by themselves are really answering the questions that I had with a particular data set that came into my hands a few years ago. So what I wanted to do, and what my group wanted to do, was develop a method that integrates multimodal data, and in my case, that means transcriptomics, proteomics, and phosphoproteomics, and infers pathways and signaling events from that data. And I also wanted it to be able to perform differential analysis between experimental groups. So here's an overview, basically, of what that method looks like. We are first going to create a list of potential paths, ligand, receptor, effector molecule, and then target. And then we're going to score them based on signal in transcriptomics, signal in proteomics, and phosphoproteomics. And then we're going to perform a kinase inference process that might poke out some interesting information about what's actually going on in this path, which is sort of a fragment of a pathway. In order to do this, I worked in collaboration with the author of XFinder, which is a method to infer ligand to target paths between cell types, or from like an external signal to your cell type of interest. Basically, how this works and how my method works is that you have an input database of known interactions from ligand to receptor, receptor to effector molecule, and then effector molecule to a target. Transcription factor might be an example of an effector molecule. And then you combine that with input data, your input transcriptomic data, to basically predict which of these paths are actually active in your sample. And so what we've basically just start off with this as the skeleton to the method, and then we extend it to include proteomics and other data. And INCIDER sort of roughly stands for inference of cell signaling transcription. So the first step in this method is to select input genes, because the paths that the method is going to spit out basically are going to consist exclusively of these input genes. So the selection of them really affects what your results are going to look like. So the strategy for this right now is to look for input genes which are highly expressed differentially expressed between conditions, differentially expressed between cell types, highly expressed in any particular cell type, and then also differentially expressed in proteomics, just to get everything possibly interesting in there. And then so we're going to make a list of possible paths from this input gene list combined with our database of known interactions, and then we'll score those. So how we score these paths from the transcriptomics data is we basically find a probability that each individual interaction, so like the ligand to the receptor, is actually occurring. We're calculating that probability using the Hill equation, which will use basically the level of expression in the transcriptomics data to figure out the sort of probability of binding at any given point. And then we take the product of all of these interactions probabilities, and that's going to be our path weight. From the path weight, since we want to do a differential analysis, we calculate a fold change between the path weights between two conditions. And then from that fold change, we calculate a score, which basically uses this evaluation function to flatten the fold change so that it exists between -1 and 1. So a score of 1 basically means that this path is more highly enriched in our first experimental group, and a score of -1 means that it's more highly enriched in our second experimental group. The second step, or the third step, I suppose, is to basically simulate a debulking of the proteomics data. So we'll take the distribution of signal for a gene in the transcriptomics data in our cell type of interest in our sample or in our group, and then we take that distribution and apply it to the bulk proteomics or phosphoproteomics signal to basically split it out. And then we scale it by cluster size in order to simulate basically like a single cell proteomics result. This relies on a few assumptions which are not really true exactly, such that like RNA and protein scale together. But as a hypothesis generation tool, this is basically the way that we can approach this right now. And then the next step is to integrate in this proteomics data by calculating a fold change between the groups in our cell type of interest, and then using the same evaluation function to derive a score for the proteomics and for the phosphoproteomics. Finally, or next to finally, we are integrating our kinase predictions by calculating a list of potential kinases that are involved with any phosphorylation that we see in the phosphoproteomics data. We use the kinase library tool for this, which will take a motif sequence and give a list of ranked kinases that are likely to interact with that sequence. Any of these kinases in the top 5 to 15 are basically plausible. So what we do is we consider that they all might be valid, and then calculate our kinase score by, for every path that has a potential kinase substrate interaction within it that we see in the data, we calculate an exclusivity index amongst the substrates for that kinase. An exclusivity index is basically just, it's at 1 if that substrate is found in that cell type almost exclusively. Its levels are higher there than in any other, and 0 if it's not enriched in that cell type. And that's the score, is just the maximum exclusivity index among the substrates for the kinase in that path. And then we add that only to the path that currently, to the experimental group that currently seems to be winning in the scores already. Then we calculate the final score by adding together the weighted scores from the other sort of sub-modules, the transcriptomics, proteomics, phosphoproteomics, kinase score. And as an addition, we also do a permutation test where we shuffle cell type labels, calculate the path weights again to see if the results that we got when the path labels were correct are actually reliable. Calculated p-value. So this is the data set that started this whole exploration. It was collected a long time ago by someone in my lab, and she hadn't really known what to do with it, with the results, but it's basically she's injected mice at different time points with cancer cells and was very curious about the development of the cancer along these different time points. She collected both single-cell RNA-seq and proteomics data, phosphoproteomics data as well from this. And what the specific question that we wanted to answer was, what are the interactions going on in the tumor microenvironment? Are there any cell type specific pathways, signaling events that we can infer that might be responsible for different rates or different time points along cancer development? So we've used this method on this data We're developing also a visualization dashboard for this that allows you to go from an overview of everything that's generated from the method to more specifics. So this is the overview of all the paths that are discovered. And then here, if you zoom in on particular paths of interest that have like high path weights, high scores, you can find some interesting examples for potentially future investigation, like this one here. We're very curious about looking at further. I also wanted to mention that when you're adding in the proteomics data, that dramatically increases the number of reliable high-scoring paths that you see. And it also increases, there's a lot of paths that might not have been really interesting looking if you were only looking at the transcriptomics data, but become interesting once you add in proteomics, phosphoproteomics, and kinase inference. One thing that we've done to sort of confirm that the paths that we're seeing are reliable is to look at their overlap with known pathways. Right now we're looking at the overlap with wiki pathways data sets, and we do see that among the paths that we identify, there's like a good overlap with pathways that look, known pathways that look relevant, such as this very large data set, cancer pathways, and so on and so forth. Next, we're also just now starting to look at a data set of COVID patients, some patients with COVID, some healthy controls. We are also picking out some overlap with very particular COVID signaling known canonical pathways, which is encouraging. And in particular, we're looking at this specific set of pathways here. We're looking to use a new method that our company has developed called IntraSeq, which is able to tag intracellular and other proteins with antibodies for investigation of cell signaling and quantification in order to validate that these interactions might be happening. So in conclusion, it does seem to be that you can get some really interesting results from this. The integration of the proteomics data does seem to really change what you see in comparison to just looking at transcriptomics alone. And it seems to be an interesting tool for hypothesis generation. There is a preprint that we are working on. The code is not yet available, but it will be soon. If you get in touch with me, or if you just Google this, hopefully in a month, it will be up on GitHub. And I wanted to say thank you to all my collaborators, in particular at UC Irvine, Chenghan He, who's actually the author of the XPointer paper and who's implemented all of this code. So yeah, thank you. Any questions?
Thank you, Claire. Fantastic presentation. Any details you can share more about the experimental data set and performance and infrastructure?
Sure. So for the experimental data set, it's mice were injected with MC38 cells, which is a model for colon cancer, and then they were sacrificed at either 10 or 14 days. For any of these samples, they would have developed some tumors. So we were looking at, there's like a very large range of tumor sizes and stages of development amongst these samples. And then for like the performance of the method, it does take a long time because we are looking at basically any path that could conceivably exist from your input gene. So if you have like a long list of input genes, it can take up to like 8 or so hours. We've done some performance optimization, such as like letting paths that have low scores drop out very early in the process. But it does still take a while, yeah.
Great presentation. My question is also about the tumor model. It's known, I think, the metabolic pathways in the tumor microenvironment, both for tumor, immune cells, they change. I think here you are doing a bulk tumor profiling, right, with the phosphoproteomics and so on and so forth.
Yeah, I think we're
like including all the microenvironment. And
yeah, we're including like a lot of immune cells,
sorting.
epithelial cells, yes.
I was wondering whether, your pathway analysis doesn't jump out, I think, but are there metabolic changes as early as like 4 days, 14 days or so in the tumor microenvironment, or
I actually have no idea.
idea.
Yeah, that's an interesting question. I'm less strong on the background of the biology of this, but
yeah, it might be early on, but yeah, I mean,
Yeah.
it's something that it's important for also response to tumor and relapse.
Yeah, thank you. Yeah.
Now let's thank our speaker.